Concept of osmosis and its role in plant cells. 1. Osmosis defined. Osmosis is the movement of water molecules across a selectively permeable membrane from an area of lower solute concentration to an area of higher solute concentration. In other words, it is the passive movement of water through a semipermeable membrane, such as a cell membrane, to equalize the concentration of solutes on both sides. 2. Selectively permeable membrane. The cell membrane is selectively permeable, allowing certain substances to pass through while restricting others. In the context of osmosis, water molecules move freely through the cell membrane, but the movement of solutes, ions or molecules, may be restricted. 3. Tonicity. Tonicity refers to the concentration of solutes in a solution and how it affects the movement of water. There are three main types of tonicity: isotonic, hypotonic, and hypertonic. Isotonic. The concentration of solutes inside and outside the cell is equal, resulting in no net movement of water. Hypotonic. The concentration of solutes is higher inside the cell than outside. Water moves into the cell, causing it to swell. Hypertonic. The concentration of solutes is higher outside the cell than inside. Water moves out of the cell, causing it to shrink. 4. Osmosis in plant cells. Osmosis is crucial for the functioning and survival of plant cells, as water movement influences turgor pressure, cell shape, and overall cell health. Turgor pressure. In a hypotonic environment, lower solute concentration outside the cell, water enters the plant cell. The central vacuole in plant cells, filled with cell sap, a solution of water and solutes, swells, exerting turgor pressure against the cell wall. Turgor pressure provides structural support to plant cells, preventing wilting. Plasmolysis. In a hypertonic environment, higher solute concentration outside the cell, water leaves the plant cell. The cell membrane pulls away from the cell wall, leading to plasmolysis. Plasmolysis can be reversible if the cell is returned to a hypotonic environment. Water uptake. Osmosis is responsible for the uptake of water by plant roots from the soil. The concentration of solutes in the soil is lower than in the root cells, creating a hypotonic environment that facilitates water absorption. Guard cell function. Osmosis plays a role in the opening and closing of stomata on plant leaves. When guard cells take up water and become turgid, they swell, causing the stomatal pore to open. Conversely, when they lose water and become flaccid, the stomata close. 5. Root hair cells. Root hair cells are specialized cells in plant roots that enhance water absorption. The root hair cell wall and cell membrane act as a selectively permeable membrane, allowing water to move into the cell through osmosis. The concentration of solutes inside the root hair cell is higher than in the soil, creating a hypotonic environment that promotes water uptake. The absorbed water then moves across the root tissues and eventually reaches the xylem for transport to the rest of the plant. 6. Osmotic potential. Osmotic potential is a measure of the tendency of water to move from one area to another due to osmosis. It is influenced by solute concentration. The more concentrated the solutes, the lower the osmotic potential. Osmotic potential is particularly relevant in understanding water movement in and out of plant cells, impacting turgor pressure and overall cell health. 7. Practical implications. Farmers and gardeners use knowledge of osmosis to manage irrigation practices. Providing an appropriate balance of water and nutrients helps maintain optimal osmotic conditions for plant growth. Understanding the concept of osmosis in plant cells is essential for grasping fundamental processes related to water absorption, turgor pressure, and overall plant health. It highlights the dynamic interplay between cells and their environment, emphasizing the importance of maintaining suitable tonicity for proper physiological functioning in plants.